Today we will continue looking at the subject of antibodies and B-cell development, and we'll finish the lecture with a summary of the key points from the slides that you have been given. We'll look at the genetic basis of antibody diversity, and under this heading we'll consider combinatorial diversity through VDJ somatic recombination in the variable domains. Here are the variable domains of light and heavy chain antibodies. Both of them undergo somatic recombination events to generate a huge amount of variability in all of these complementarity determining regions here. In addition, we'll look at something called junctional diversity, and this I'll explain once we get to that point. It's another aspect of diversity. We'll also look at something called class switching. We've talked about the classes of antibodies. Initial antibodies are IgM, and then we have class switching occurring to IgG, which is a much more stable and effective antibody in the longer run. And while this is happening inside the B cell, the antibodies are experiencing, or the genes that code for the antibodies are experiencing somatic hypermutation within these regions here. And finally, or actually these two subjects will be interspersed to an extent, we will also look at the stages of B-cell development, coming from immature to a mature B-cell. And we start with a pro-B cell and a pre-B cell, then transitional B cells divided into T1 and T2 B cells, before finally having a mature B cell. And the mature B cell is actually still considered naive, and the naive B cell needs to be activated. We've actually already looked at the process of activation. This is where B cells are exposed to antigen in the lymph nodes, and then they become activated and undergo clonal proliferation and turn into either plasma cells, which are antibody production factories, or memory B cells, which give a long-term memory for a secondary response at some later time. So we've looked at activation, but we will look at these prior stages from the pro-B cell through transitional to the mature naive B cell. So, okay, let's make a start with the genetic basis of diversity. The variable domain of the heavy chain locus is made up of a V, a D, and a J segment. Downstream of that are the constant regions. And the gene here looks very, very different to the final gene in the transitional B cell. Why is it different? In the transitional B cell and downstream from that, the mature B cell, there is one V segment, one D segment, and one J segment. However, initially, there are 40 V segments in the genome of the B cell at this locus on chromosome 14 for the heavy chain, 23 D segments and 6 J segments. And what happens is that recombination occurs between these in order to select one J and recombine it randomly with one D. So any one of these 6 J segments may recombine with any one of these 23 D segments. So if you remember from simple combinatorial probability, that means that we have 6 multiplied by 23 different possible combinations that we can get from this. Subsequently, another recombination event occurs in order to recombine the DJ recombination product with any one of these 40 segments. So you could take your calculator out and multiply 6 by 23 by 40, and you have a huge amount of possible combinations. And it's this process of generating combinatorial diversity which gives B cells the capability to produce any one of a huge number of possible different antibodies in order to cope with the huge vast array of possible different pathogens that may infect humans at some time. Now we come to the light chain, and a very similar thing happens. We have J segments and V segments, but none of these green D segments. So the heavy chain has V, D, and J segments. They get recombined with the constant domains. For the light chain, it's V and J segments that get recombined and then recombined with a constant domain. There are two differences. One for the light chain is the lack of any D segments, and secondly, the unusual thing that for each J segment, it has its own constant segment attached, whereas in the heavy chain, there's a single constant region, and all these J segments are individually capable of this recombination. Here, the constant and the J come as a package, so you can have J lambda 1, C lambda 1, J lambda 4, C lambda 4. For example, obviously between these there is 2 and 3, up to 5. So this is VJ recombination as opposed to VDJ recombination. The term lambda is referring to the fact that there are actually two different loci for the light chain. So one locus on chromosome 22 is for Ig lambda, and on chromosome 2 is the kappa locus, this Greek letter kappa here. And that is similar, the kappa locus is similar to the lambda locus in the absence of any D segments, no D segments here. But the J segments are organized in a similar way to the heavy chain. So the J segments are all together. There are no associated constant domains. There's just one constant or constant region downstream. So this arrangement is similar to what we find in the heavy chain. In both cases, there are no D segments. And likewise, we have a number. It's not the same number. It's different, but there are about 30 lambda B segments and 4 J segments. For kappa, 5 J segments and about 40 V segments. The L refers simply to a signal sequence which is necessary during the translation of this protein to direct the antibody to the endoplasmic reticulum, the rough endoplasmic reticulum. So the next slide, we look at the molecular process by which the recombination occurs. So I hope that you've got the concept now that we've got a lot of diversity here, all these combinations, and it's a fairly random process for the light chain, whether we have lambda or kappa. This will become even more clear after we look at the molecular process of how the recombination occurs. So this is somatic recombination in B cells, not germline recombination. Attached to each of these segments, in fact flanking each of these segments, are recombination signal sequences, and these are basically composed of unconserved spacer, and on one side a heptamer, and on the other side a nonomer. Nonomer means 9, heptamer means 7 nucleotides. And there are two types, one with a 12 base pair spacer and the other with a 23 base pair spacer. So the sequence doesn't matter here. However, the size does. So here's an example. This is, as you can see, the heavy chain. It says H, but you'd know it was the heavy chain because only the heavy chain has D segments. So each V segment has a downstream 3' spacer. Each J segment has an upstream 5' spacer, and each D segment has an upstream and a downstream spacer. The light chains also are flanked by downstream and upstream recombination signal sequences. And an enzyme, or an enzyme complex called RAG1, RAG2, which actually has two RAG1 proteins and 2 RAG2 proteins assembled together, and the DNA wraps around this enzyme, and those spaces are recognized, and these different sizes of the spaces determine what's going to be recombined with what. As for the steps in the process of the recombination, so this is going to involve physically bringing segments together and also cutting the segments. This will lead us on now to the second mechanism for generation of antibody diversity. So the process of recombining the V and the D and the J segments actually also introduces at the junctions some other random mutations and changes, and which then greatly increases now the diversity. So we call that junctional diversity. So here we have the mechanism of VDJ recombination, or actually what's shown here is V and J, so this is for a light chain. Really, I want to simplify this for you. The thing which I want to draw your attention to on this slide is that once the DNA is cut around these signal sequences, which are represented here, this is going to be the intervening space which will be thrown away basically, and here will be the two ends of a J and a V segment that need to be joined together, resulting in this. So here we've joined together two segments, a V and a J, on one of the light chains. This little red thing here represents something a bit strange. When DNA is cut, say by a restriction enzyme, it leaves two free ends, a 5' and a 3' end. However, here what happens is a esterification reaction occurs in order for the 3' end of one strand, DNA is double-stranded, to be esterified to the 5' of the other strand. So, and this connection here is called a hairpin loop, so the DNA is closed. The next slide is going to describe what happens at that hairpin loop, and the process by which these two ends get joined together after the hairpin loop is formed will generate the junctional diversity. So here we have our hairpin loop, and just by way of example, a few amino acids at the end. So here we have one segment, here we have another segment. The end is cleaved, and it's cleaved asymmetrically, meaning not symmetrically. So we've introduced the cell, the B cell has introduced this transesterification here and joined these two together. And after that, then this is cut, but it's not cut here, it's cut somewhere along here. And that's random. So in the slide, it's it's cut 4 nucleotides away from what was the end, but it could be 3 or 2 or 1, it could be anywhere around this end. So once we have a cut in this example here, just before that T residue, then it means that what we're going to have on this end here is AGGT, and in red here ACCT. Where did the ACCT come from? Well, it's here, TACCT. So we've got this here, and up here with same type of story. We have CC, TC, CC, TC, and then after that is a GAGG, GAGG. And these are called P nucleotides, palindromic nucleotides. You see, here was the end, supposed to be here was the original end, and here was the original end of the other segment. But now we have a lot of extra nucleotides that are a result of that asymmetric cleavage event up here. And those are called palindromic because there's a symmetry, isn't there? This ACCT is the reverse complement of this going backwards. So A pairs with T, C pairs with G, again C pairs with G, and here this final T was originally pairing with this A, as you can see up here. So these are not random nucleotides. These nucleotides are related by a type of symmetry to these, and we call those palindromic, or for short, just P nucleotides. So the random aspect here generates some degree of diversity now. Additionally, there's some random deletion of nucleotides that happens at the end. So we had all of this up here, and then in this example, this is just an example, many nucleotides have been deleted from this segment on the left, and all we've got now is the A and G. Here we have 2 nucleotides deleted, and we've got 6 nucleotides at this end here. And again, that's random. How many nucleotides get deleted is random. Not content with this, B cells have another enzyme called terminal deoxynucleotidyl transferase, TDT, which actually randomly adds nucleotides and those are indicated in blue here, and those are called N nucleotides. So we've got P nucleotides and N nucleotides. So all of this randomization happening here now gives, for any given V and J segment or D and J segment, if it's a heavy chain, this now gives us, on top of the combinatorial diversity, a huge amount of added diversity. When I say added, actually probability is multiplicative. So this is hugely expanding the overall diversity possible in the variable domain. Then finally, also some nucleotides may get removed from there. There will be along here some area of matching, and in this case, in this example, there's a T and a G and an A and a C that they match up. So there's obviously not a complete match, but there's a small region within this that can match up, and then the gaps are filled in, which again is kind of a random process. So now we've got all of this. So we should have, if it wasn't for the junctional diversity, we'd have AG, and then we'd have a G here rather than this T. And then we'd have and I'm basically just reading off what's up here and comparing with what's down here, which is so different. So you can see that around here at this junction now we have something different. It might occur to you, and it should occur to you, that with all this random process, the reading frame is not going to be preserved. If you remember that in order to have a string of amino acids added by the ribosome as it reads messenger RNA, which will be transcribed eventually from this genetic locus, then everything all needs to be in frame. Every 3 nucleotides is a codon, and each codon codes for an amino acid. But here, after we've finished this segment now, there's a chance, in fact there's 2 chances out of 3, isn't there? If there are 3 nucleotides might have been added or deleted in this whole process, so we would have maintained the reading frame. However, the reading frame may not be maintained. So that means that not only do we have variation here, but everything downstream from here may be different because of that. So this is a huge amount of variability, which is a good thing, which is what we want. We want to have B cells with the capacity to mature into a population of cells capable of expressing a whole almost infinite range of antibodies that could detect any possible pathogen that may be present on the planet now or might be present in the future. So that's good. However, that also presents a problem. Not all of these are going to work. Not all of these are going to be functional. Some of these are going to completely change the structure. You saw about the immunoglobulin fold that there's a domain-like structure. Not every one of these alterations is going to give us a functional antibody, especially if we have things that are out of frame and the wrong reading frame. So how do we get rid of those useless antibodies that simply would lack the capacity to fold up into a functional? When I say what I meant was, how will we get rid of those useless genes or gene rearrangements that would fold up into a useless antibody? And that's what the next slide will look at. And that brings us into now the idea of the B cell development. And going from a stem cell, we'll look at going through stages of the pro-B cell and then to the pre-B cell, before we get to the immature B cell, which will be the transitional B cell. So when we look at this stage here, all of this is happening in the bone marrow, and after we've reached this stage, then the B cell is able to be released from the bone marrow, and well, I'll tell you what happens after that when we get to it later. So looking at the heavy chain, what's going on, so the different cell stages, and then what's happening to the heavy chain, what's happening to the light chain, and what's the overall physical status of the protein and the immunoglobulin that's produced. So to start with, we have there's been no recombination. This is a stem cell, and there's obviously no immunoglobulin. Once we enter the pro-B cell stage, the heavy chain DJ rearrangements are occurring. Nothing's happening with the light chain, so we've got no antibody because we need heavy and light chain. But also we need to have the D and the J segment after that's been recombined now has to recombine with a V segment, and that's in the late pro-B stage. And still nothing's happening with the L chain, and so we've got no mature immunoglobulins produced. Then we have the pre-B cell stage, and pre-B cells are larger than the following stage, which are, so we define large and small. So we had early and late pro-B cells, and when we get to the next stages, we have large and small pre-B cells. So once we enter the pre-B cell stage, all the VDJ recombination on the heavy chain has been completed, and yet nothing has happened in the germline stage. But something has changed here, and that is the heavy chain is made. And this Greek letter mu, if you remember, is referring to the fact that the constant region will be IgM. So an actual antibody is now being made. The heavy chain is recombined, and a heavy chain protein, immunoglobulin, is actually being made. After that, then we have the VJ recombination occurring on the light chain. And the heavy chain, this mu chain, actually makes its way to the endoplasmic reticulum. After the rearrangement of V and J segments, then we have physically production of heavy chain as well as light chain. And remember that we have two possible light chains. It could be lambda or it could be kappa. And the immature B cell is now expressing IgM on its surface. Now coming back to this stage where we start to have this heavy chain being made, basically a testing step has to happen here. And in dark blue here is shown the heavy chain. And the B cell actually has a cellular mechanism to do a quality control check to see if the rearrangement has produced something that's functional. And the way it checks this is the B cell produces another chain called a surrogate light chain. This is the composition of it. You don't need to memorize that. So a surrogate هو بديل يعني زي sorry if I talk in Arabic now. زي الأم اللي ما لهاش حليب وتيجي تاني بالرضاعة. So this is tried in Arabic to illustrate the concept of surrogate. So this is not actually the light chain which is going to make up the antibody. This is something that doesn't undergo any V or J recombination, but it's basically a tester. And if this is able to associate properly with the heavy chain, and if the heavy chains are able to dimerize through these disulfide bonds up here, and if the heavy chains are able to associate with some co-receptors here, which we haven't talked about, but there's a co-receptor needed for antibody engaged or B cell receptor engagement on the cell surface. If the heavy chain that's made is able to interact properly with the co-receptor, it's able to embed itself into the membrane of the endoplasmic reticulum where the co-receptor is, and if it's able to associate with these surrogate light chains, then that's a signal that everything is good, that this pre-B cell is now ready to work on the light chain. If it's failed, then there's no point to work on the light chain. At this stage, once we have a successful heavy chain, then the pre-B cell, the large pre-B cell, undergoes rapid proliferation. It makes many copies of itself. Something works, the heavy chain works. The light chain rearrangements might not work. So if we have many of these pre-B cells, all with an identical functional heavy chain, then it means each one of them has the opportunity to undergo some VJ rearrangement in the light chain and some of those will work and some of those will not work. Imagine if we had just one cell and its first attempt to make a light chain was a failure. So that successful heavy chain would be lost and wasted. So in order not to have any wastage, then the pre-B cell makes many copies of itself, and those undergo all of this light chain arrangement. And finally, we should end up with an immature B cell. The receptor is mature. I added that up here. So this B cell is not mature, but the receptor is mature. So we have an immature B cell, and this one has a fully functional now B cell receptor. The difference between the B cell receptor and the antibody is simply this little cytoplasmic tail, which after a B cell finally differentiates into a plasma cell, very late after activation in the lymph nodes, then by alternative splicing, this cytoplasmic domain will be excluded, and we'll only have the extracellular portion, which will be secreted as soluble antigen. So what happens next? We've got the immature B cell, and it's expressing a mature B cell receptor. There is an issue, and the issue is that all these huge array of many different B cells, all with a unique B cell receptor, which is able to associate on the membrane with light and heavy chains together. It seems to be a good functional B cell receptor. Some of these B cell receptors will recognize self. They'll recognize our own body, and that will be harmful to us. So there needs to be a screening process, and we call that negative selection. There needs to be a screening process to get rid of any self-reactive B cell receptors. And actually, it's not the receptor that is gotten rid of, it's the B cell that produces that receptor. If that receptor is self-reactive, then the B cell that makes that receptor will be deleted. And this negative selection at this stage is called central tolerance. It's called central because it happens in the bone marrow. And tolerance, it's because the result of this checking process means that our B cells tolerate self. They don't react against our self. So central tolerance screens out any B cells that might react against self. So here we have an example of a B cell expressing IgM, and it's in the bone marrow. In the bone marrow, there are opportunities for every B cell, every immature B cell, to react against self-antigens. If they don't react against self-antigens, it means there's a lot of hope for that B cell, and that B cell can then enter the bloodstream. And at that stage, the B cell actually is also expressing IgD. So IgM and IgD are expressed on the surface. IgD does not seem to be significant as an antibody. Its relevance seems to be for some type of signaling or monitoring process as a receptor. IgM, however, will be secreted from a mature activated B cell. That's coming later. So the B cell has undergone this process of negative selection, and everything's fine with it, so it can be released to the blood. However, if a B cell does react with self-antigens in the bone marrow, then that B cell is not released. In fact, it will undergo, in most cases, apoptosis. Those B cells that are released, they're still not fully mature, and they're called transitional B cells. Transitional B cells. They still need to undergo some negative selection, because not all self-antigens are going to be present in the bone marrow, and there are some antigens, some self-antigens, which are only expressed in very specific places in the body. So human B cells are given a chance through various locations to interact with self-antigens and be selected against, and we call that peripheral tolerance. If you look in some textbooks, you'll see referring to the fact that peripheral tolerance is happening in the spleen, and that's definitely true in mice. In humans, it seems to be much more complicated, and negative selection or peripheral tolerance seems, yes, to happen in the spleen, but also there seems to be many other locations where this screening process is happening. So we have central tolerance and peripheral tolerance, where negative selection is happening. Once the cell has passed this stage and enters the lymph nodes, then it's a mature B cell. That means it's all ready to go. But it's still naive. Naive, it means in Arabic, savage, because it's never seen anything bad before. It's lived a very sheltered life with its mummy and daddy. It's only seen friendly self, and it's passed those checks. So it's still naive. It's not been exposed to antigens from pathogens. But it's mature because it's expressing functional active B cell receptors ready to be activated and ready to turn the B cell into an antibody producing factory. Now, I just want to talk about the epitope for a few seconds, actually, but it's a simple terminology that you should know. That the epitope, that's what binds to the paratope of the antibody. The epitopes may be linear, meaning that the antigen which contains these amino acids epitopes are in a linear stretch, amino acids next to each other. However, we can also have, and most epitopes, most B cell epitopes are like this, discontinuous, meaning that as the protein folds, amino acids that are physically close to each other in the folded protein are actually maybe far away from each other in the sequence. So 1, 2, 3, 4, 5, this amino acid number 6. The second amino acid that's recognized is 7, 8, 9, 10, 11, 12, 13, is number 14. So amino acids 6 and 14 are recognized. So they're discontinuous. This is continuous. Amino acids 2, 3, and 4 are recognized. So that's a kind of a side note about terminology of the epitope. Now coming back to the antibody. We've looked so far now at the generation of diversity, combinatorial diversity, junctional diversity in the heavy and light chains, and we've seen how this relates to various stages of the B cell development. Now we need to look at after activation. So we had the naive B cell, which is mature but naive in the germinal center or lymph nodes. Finally, it's found itself an antigen from something pathogenic that it can recognize. Then it's going to become either a plasma cell or a memory cell. But after that, something else can happen, and that's what we're going to talk about in this slide. There are only 2 slides left, this slide and the one following it. And that something else is the class switch recombination. So the first antibody which is going to be made is IgM, that's that pentameric antibody. After that, we have IgG. And some of these antibodies have subclasses as well. I remember with the term class or isotype, I used synonymously or interchangeably. So here we've got the VDJ, so this is the heavy chain. So we're not interested in that now. We've talked about that. What we're interested in are these constant domains symbolized by the letter C. Remember, we have IgM and IgD expressed on the surface of the immature B cell, and this one mu will make IgM. Downstream we have gamma, so that's going to be IgG3. Here we are going to make IgG1, gamma 1. Of course, there are things I'm not mentioning yet. I'm just looking at these grey boxes. Here we have IgG2A. Here epsilon, that would be IgE. Alpha, that would be IgA. So each of these different types of constant domains, which will make these different classes, they give different effector functions for the antibody. Remember, we talked about the Fc receptor, the neonatal Fc receptor, complement C1q that's able to recognize antibodies. Many different effector functions are possible with these different constant regions. And the little oval round type of shapes here simply recognize switching elements that are able to recombine with each other in order to delete things in between. So if we started off with IgM being produced and have you know that we have introns and exons, so we can have all types of things here, it doesn't matter. Once this DNA becomes transcribed, it will transcribe this V and D and J, and then loop out here, and we'll have this constant domain in there, and then that will be translated into viable proteins. So the translational machinery won't see this this domain, because this will be spliced out inside the nucleus of the cell. But these signals are recognized in the nucleus of the cell when it's time to switch from, say, IgM to IgG2A in this example. So if this is the constant domain we want in order to have the effector functions of IgG2A, then the switching region here, which associates with that, will recombine with this in order to lose everything in between, which is down here. That's all going to get thrown away. And now what's left is, after the VDJ, and got the going in the intron here, and the next exon is gamma 2A, which is what we wanted. So we lost everything in between, and when this is transcribed, we'll end up with a functional antibody containing the variable VDJ domains and just the constant region here to make mRNA, and then the final antibody. Now we have just one more slide. All we're going to talk about here is somatic hypermutation. I've mentioned it before. It occurs in the germinal center, which is here within the lymph node. And the very new thing is that the enzyme that mediates the somatic hypermutation is the same enzyme which is important in the class switching that we looked at in the previous slide. Now, somatic hypermutation was identified when people started to look at B cells over a period of time, and looking at many different B cell clones. So here represent different B cell clones, and sequencing them, and finding them, not surprisingly, they're all the same. Before the class switch has occurred, where we have just IgM, which is the first antibody class produced. So there are lots of B cells. There was proliferation of an activated B cell, and so there are lots of B cells all with the same sequence. However, after the class switching has occurred, and we get IgG, people notice that when they sequence clones, they find lots of mutations. So each of these orangey circles here represents a mutation. And those mutations, not always, but they, to some high degree, they correspond fairly closely with the complementarity determining regions that we looked at earlier, which are in these libs. Perhaps here it's better than for CDR1. So the first strange thing is, it's not strange now, but it was strange when it was discovered, is that it's not that after all this amazing combinatorial diversity through the VDJ recombination, along with the junctional diversity with the P nucleotides and the N nucleotides, in addition to all of that diversity, after class switching has occurred from IgM class to IgG class, more mutations appear here. And it's called somatic because it's in the B cell, which is a somatic cell. All the cells apart from the germ cells are somatic cells. And somatic hypermutation in humans has only been identified in lymphocytes. Okay, so somatic hypermutation. So we get even more diversity happening here. And I may remind you, when we looked at the anatomy of the lymph node and the germinal centers, that there's actually a process of improvement happening. So all of these different clones here with some mutations in, basically all competing against each other. And there's a backwards arrow as we go from dark zone to light zone and then back again, starting with a naive cell up here to the fully mature plasma or memory B cells here, before finally preparing all these amazing effector B cells then there's a kind of cycling backwards and forwards between the dark and the light zone, where different mutated forms get tested against each other, and the best ones win and finally get promoted to become a plasma cell or a memory B cell. And somatic hypermutation can improve the antigen affinity of the B cell receptor by several orders of magnitude, up to about 1,000-fold improvement. And all this occurs after about 2 weeks, which is about the time, 10 days or so, where we have this switch from IgM to IgG. Some of you who are interested in laboratory and diagnostic things may know that detection of IgM or IgG is used to indicate when infection occurred, if it's a very recent infection or an older infection, but that's another story. Now the enzyme, finally, the enzyme that does this is called activation-induced cytidine deaminase, and this enzyme is important for the class switch recombination, which was the previous slide that we looked at here, and kind of repeating myself, I guess, but just want to emphasize, and it's important for the somatic hypermutation, which is the subject of the current slide we're on. So what does it do in somatic hypermutation? Well, when it finds a cytosine, it's able to deaminate the cytosine and turn it into uracil. And DNA is not supposed to have uracil, and so this is an error. And when this error is corrected, it's commonly replaced with an A, adenine, and thus, once this chain is produced during a replication, then on the other chain we're going to have T. So we started with CG, and we end up with AT. And that's the basis for somatic hypermutation induced by activation-induced cytidine deaminase occurring during the time of the class switch recombination, in order to make the most effective antibodies that are possible. And that brings us to the end of this lecture. And next we will look at, in the next lecture, we'll start looking at the T cell. Thank you.